Moving on to sensory systems and plants. This is part 2. And this is more about gravity. Gravity. So, I mean, think about plants. Where did plants come from in the first place? Well, probably green algae, which exists in water. So there was a move to land, and the first plants were mosses, still pretty tied to water. And then ferns, and then some gymnosperms and angiosperms, which are the flowering plants, were the last kinds of plants to evolve, but they're the most numerous plants. And so, but now they're not in the water anymore. So nutrition has to be derived from air and from soil. So roots get nutrition from soil, and the shoots get nutrition, carbon dioxide, from the air. So how does the plant know which way is up or down? How do plants perceive gravity? Well, we know that roots grow down. Roots grow downwards. And we know that shoots grow up. This is gravitropism, movement of a plant in response to gravity. So positive is with gravity, negative is against gravity. But where then is gravity perceived by the plant itself? Well, our good friend Darwin, he removed the root cap. Roots have a root cap. And the root cap's purpose, at least part of its purpose, is to protect the growing tip of the root from the friction of the soil. So there's the root cap. You remove the root cap. It's made of very tough protective cells, but it does wear away as the roots go through the soil. When the root cap was removed, the root no longer responded to gravity. So the sensing of gravity has something to do with the root cap. How can we refine it? Well, Darwin just simply cut off the root cap. It turns out there are cells, however, just under the tip of the root cap that sense gravity. It's not the tough outer cells, but it's with further experiments, removing some of the tough outer coating, it turns out that the inner cells are the ones that detect it. What is the mechanism? Well, there's a couple of hypotheses for the actual mechanism of how it happens. Plant cells have structures known as amyloplasts. They're like vesicles, and they store starch. So plants, of course, they store starch, and we eat plants for the starch so that we can get the broken down starch sugar that we use for cellular respiration. The starch in this case is amylose. You can see that they contain starch because you add iodine. So iodine is a stain that reveals the presence of starch. It turns the starch blue, kind of a blue-black color, so you can always tell if starch is present. So this is one hypothesis called the statolith hypothesis. So some root cells have statoliths, which are really quite large amyloplasts. So they contain starch, they're quite large, so they're relatively heavy, and they're pulled to the bottom of a cell by gravity. Gravity pulls them down in this direction, as you can see. And it activates pressure sensors in the membrane. So maybe these are pressure sensors. And depending on the sensors that are activated, the roots will grow down with gravity, positive gravitropism. This is the root cap here. These are the amyloplasts inside here. So you can see where they gather. Which way is down? Well, it must be this way because all of the amyloplasts are on the side. And roots grow down. How does a lobster know which way is up? Why are we talking about lobsters? Well, grains of sand in the antennae of lobsters are called statoliths. And that's how the lobster knows which way is up. Some plants don't have amyloplasts, but they still respond to gravity. It's called a gravitational pressure hypothesis. So instead, there are vesicles within the plant called integrins, and they're attached to the cytoskeleton. And the cytoskeleton is attached to receptors. And when these fall with gravity, they pull the receptors down. So that's thought to be the gravitational pressure method or mechanism rather. And the receptors on the other side, these ones here, are compressed with pushing. So as you could imagine, a botanist sent seeds into space to see what would happen without gravity. Plants also respond to touch. Have you ever seen a mimosa plant? A mimosa plant? When you touch the leaves, they just go like that. It's very neat, actually. I'll try to find a video to show you. So plants do respond to touch. You can just touch the mimosa leaf and all of the leaves that are on that particular stem will close. They'll just close right up. Plants, if they're prodded a lot, will get shorter and stockier. What is the mechanism of that? It turns out genes are turned on. The cell walls get thicker so that they are more resistant to touch. So wind, for example, is a form of pressure that plants experience. So the perception of an environmental signal, signals are such things as gravity and wind, touch pressure, and light. Well, how, after perceiving an environmental signal like that, does the plant respond and why? So there must be some kind of transduction from that external stimulus to an internal signal of some sort. That is a cellular response. And how does that occur? Well, like Stefano was saying, there is an electrical component to that electrical signaling. So maybe you've taken some nervous system classes for animal nervous systems and know that neurons are very negatively charged on the inside, positively charged on the outside. And that polarity is caused by sodium, mostly on the outside of the cell, and large negatively charged proteins and chlorides on the inside of the cell. So it's similar, but not exactly the same with plants. There is electrical signaling or depolarization within cells. So in this case, the positive outside of the cell is maintained by hydrogen and inside by chloride and some hydroxyl ions. And there is a charge difference. With animal cells, it tends to be minus 70 millivolts. With the plant cells, it's minus 150 to minus 200 millivolts for plant cells. And they are a little bit like nerve impulses. So they do travel from cell to cell, although not cells that look anything like neurons. They have receptors. In the Venus flytrap, there are receptor cells and hairs that stick up. Here are the hairs. And what's very interesting is that when an animal, usually a fly or some other kind of animal, I think that slugs or snails are not as likely to be trapped by a Venus flytrap, but when two of these hairs are touched by an animal within, I'm trying to remember what David Attenborough said in his video, within, I think it's 20 seconds, then the Venus flytrap will close. It will snap shut on the insect. Yeah. And that occurs because of osmosis. So water rushes into the cells that are responsible for closing up the two sides of the Venus flytrap. The cells just swell up. They're near the outside surface. And they respond. They respond when calcium flows into the cell. So it is really quite similar to the nervous system. The trap closes in 0.5 seconds, very quickly. So that's a good example. And here's the mimosa plant. You can touch the plant. So this plant's touched there, and you can see the entire compound leaf closes up to protect it. So it is an electrical potential that occurs, a change in membrane potential. It's a little bit different than an action potential you might be used to. The membrane potential here, resting potential is -160. The voltage change may go up to 0. The positive ions in this case are potassium, not sodium, that rush into a cell. And the charge gets more positive and then goes back to normal. Rapid depolarization and rapid repolarization as well. Yeah. So good. We've looked at phototropism with light, gravitropism, movement with gravity. There's also, we've looked at electrical now, the Venus flytrap, the mimosa. We've looked a little bit at touch and wind. That collectively is known as thigmotropism. So these are directional growth responses to signals: phototropism, gravitropism, thigmotropism, and electrical signals. Awesome. So I'm going to stop there.